Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Gowani Government College, Binmar. Today, I will deliver a lecture on human blood groups. This will be my second lecture on physiology of blood. In my first lecture, I told you about the composition of blood and the function of different components of blood. Now let us discuss about the human blood group system. The human blood groups were learned from tragedies, death caused by mismatch during transfusion in the ancient time. The ABO blood types were identified in 1900 by Karl Landsteiner. He noticed that the blood of the donor would clump or clot with that of the patient, that is known as the agglutination, during the transfer of blood from one to another, while the other blood types were identified later. So therefore the Landsteiner is known as the father of blood groups. Now what are the blood type as given or as proposed by the Landsteiner? In the case of ABO system, there are presence of agglutinogens, and they are the specific glycoproteins present on the red blood corpuscles membrane as shown in this slide. All RBCs in an individual carry the same specific type of agglutinogens, and on this basis the ABO blood group system has four types of blood. One, the type A, in which the red blood corpuscles carry agglutinogen A, that is antigen A. In type B, the RBC carry agglutinogen B, it means the antigen B. The type O is basically 0, RBC does not carry any antigen, neither A nor B agglutinogens. And type AB, RBC carry both the antigen or agglutinogens, that is presence of antigen A and antigen B. Now let us discuss about the genotype and phenotype of the ABO blood types. The first one is the blood group type A, the second one is blood group type B, and the third one the AB, fourth the O or 0, the antigen A in blood group A, antigen B in blood group B, both the antigen in blood group AB, neither an A or B in the case of blood group O. As far as the antisera or antibodies are concerned, the antisera B in blood group A, antisera A in blood group B, none of the antiserum is present in blood group AB, and antisera A and B both are present in the case of blood group O. While the blood group A can donate to A and AB, B can donate to B and AB, AB can donate only to the AB, O can donate to all, and as far as they can receive the blood, A is received from A and O, B can receive from B and O, AB can receive from all, and O can receive only from the all. Now let us look at the genotype. This is the homozygous condition. Both the alleles of the allelic pair are same. This is both the members of allelic pair are different. One the dominant one, the other is the recessive. This is the dominant, both the homozygous for blood group B, and this is the heterozygous for blood group B. This is the codominance with both the antigen are present, and this is the homozygous recessive for the blood group O. So this chart actually summarizes the ABO blood group system. Now let us discuss about each blood group in detail. First, the type 1, which we have already studied, that it carries the agglutinogen A, and agglutinin B. It means the antigen A and antibody B. The agglutinins are preformed antibodies present in the blood plasma, and they bind to agglutinogens that are not carried by the host RBC. So they cause the agglutination or the clumping or aggregation and lysis of incompatible RBC. If there is mismatch, such type of condition can occur. This is the mix type A plasma with type B plasma, and the result you can see. The transfusion reaction, the type B recipient and the type anti-A donor RBCs are agglutinated here shown in this slide, and then they block the small blood vessels, the capillaries. Now let us look at the second type, that is the type B, where RBCs carries B agglutinogens and plasma carry the not the agglutinogens but carries the agglutinins. So we were talking about the type B blood group, where I just told you that plasma contain agglutinin against the agglutinogens. It means the agglutininin which is present here is not mentioned here. It is actually agglutinin A or the antibody A. Now next, the blood group O. In the case of blood group O, RBC carries neither type A or type B agglutinogens. It means both the antigens are totally absent in this case. While plasma contains agglutinin against both type of antigens, it means it contains agglutinin, that is the antibodies against the agglutinogens A and B. It means antisera A and B are present, which can cause clumping of those RBC which carries antigen A or antigen B. Therefore, the person which blood group O can receive the blood only from the person who has the blood group O. So donor and receiver both must have the blood group O type. And the last one, that is the type AB. In the case of type AB, both agglutinogens A and B are present. It means antigen A and antigen B both are present. As I told you earlier through the slide, that the glycoproteins A and B or antigen A and B both are controlled by a dominant allele, and when both the dominant allele are present in one allelic pair, both shows or both express their character, and as a result of which the blood group has both type of antigen and it becomes type AB blood. If we consider the agglutinins, the antibodies, they are totally absent. Therefore, it can receive blood from anyone. It can receive from A, B, AB or O. This is the summary of the ABO blood group system, where A has antigen A but antisera B in the plasma, and while blood group B has antisera A but antigen B, while O has neither antigen A nor antigen B but has both the antisera, and just opposite to O, the AB has both the antigen but neither the antisera A nor the antisera B. Now the second type, that is the RH factor or the RH antigen. The RH factor is another protein factor on the red blood cells. The term RH comes from the type of monkeys used to test for this factor, originally the rhesus monkey, and hence the name is given as RH, R capital and H small, for the rhesus monkey, here clearly mentioned in red color. We are the primate, and human being which are primate has Rh factor as shown in this slide. Apart from the human, all the other primate are shown in this slide, beginning from lemurs, tarsiers, South American monkeys, old world monkeys, orangutans, chimpanzee, gorilla, and gibbons. All these come under the order primates. Now, what are Rh blood group? It can be either positive or negative. It means it can contain the Rh agglutinogens. If it contains the Rh agglutinogen, it is positive, and if it does not contain the Rh agglutinogen, it is negative. 85% of people have Rh present on their red blood corpuscles, and their blood is referred as Rh positive, and they can receive Rh positive and Rh negative blood, while remaining 15% do not have Rh present on their red blood cells. Their blood is referred to as Rh negative, and they can receive only Rh negative blood. The problem with Rh negative mother and her Rh positive fetus is generally dangerous, which is shown in the future slide. Now this is the case. The mother is Rh negative, father is Rh positive. Now the first baby, if the first baby is Rh positive, which it will happen if father has both the dominant gene. And this is the case of second baby, where again the case is the baby is Rh positive. In the first case, it is protected by the plasma blood barrier. The mother is not exposed to the Rh antigen until the time of childbirth due to placental tearing. The second Rh positive fetus develops severe anemia and dies due to hemolytic disease of the newborn, which is known as erythroblastosis fetalis. And the treatment is nowadays available. There is use of anti-Rh gamma globulin to mask the Rh agglutinogens, and this injection is generally given to Rh negative female if she wants to conceive again with Rh positive male, because there are chances that the child or the developing fetus will have Rh antigen in his or her blood. Now this is the summary of what we have studied about the Rh factor. So continuing the summary, the Rh positive father and Rh negative mother may conceive an Rh positive baby, and in the first case there is no problem. The problem arises after the birth of the first child, because now mother's body has just received the Rh antigen, and the mother's body start producing antibodies against the Rh antigen. So when the second time the same female conceives, and if the developing fetus is Rh positive, so the immune system of mother acts more rapidly and more vigorously. And when the blood transfusion takes place during the pregnancy, during the development of the fetus, it causes clumping of the RBC, and the disease is known as the hemolytic disease of newborn, that is the erythroblastosis fetalis. So this completes my lecture on human blood group system. इसी के साथ मानव रुधिर समूह पर मेरा ये व्याख्यान समाप्त करता हूँ, जिसमें मैंने आपको ABO सिस्टम और RH कारक के बारे में बताया, और किस तरह से RH कारक अगर मादा के अंदर नहीं होता और नर के अंदर पाया जाता है तो कौन कौन से रोग हो सकते हैं और क्या क्या संभावना है. So this finishes my lecture. Thank you.